So in this lesson, we are going to compare and contrast the prokaryotic and eukaryotic cell. So I'm sure that most of you already know this, so it's a revision. Then we will also state the key features of viruses. So you would like to know the structure of virus, because virus play a very important role. It can cause, a lot of viruses can cause diseases. So we want to know the basic structure of the virus, which is made up of nucleic acid, capsid, and all the phospholipids. Okay, so let us look at pro and eukaryotic cell. Now, prokaryotic, the word pro stands for before, right? Karyo stands for nucleus. So prokaryotic cell means a cell before the nucleus is formed. That means this cell has no nucleus because it's prokaryote, okay? Eukaryotic cell, U means well. So there's a well-developed, karyo means nucleus. So eukaryotic cell is a cell which contains nucleus, all right? So why do they call those cells pro and eukaryotic cell? Is because it is based on the evolution of organism on Earth. So it's believed that the very first organism that's produced on Earth is a very simple organism, which is made up of the cell surface membrane and some chemicals inside the cell, like the DNA molecule. All right. So this cell is very simple. There's no nucleus in it. So this kind of cell without the nucleus is called the prokaryotic cell. And the organism that contains the prokaryotic cell is called the prokaryotes. Okay, like bacteria are prokaryotes. It is made up of prokaryotic cell. So as the time passes by, there start to be some compartmentalization in the cell. So compartmentalization in the cell means that in the cell, there's some specialized structure that's being formed inside the cell, like nucleus is being formed. And then you've got the rough ER and smooth ER being formed, and you've got the Golgi apparatus being formed, okay? How to draw Golgi apparatus being formed, and a lot of other organelles being formed. So this is called compartmentalization. That means you divide the cytoplasm into different compartments using maybe membrane or other molecules, all right, to form that organelles. So the organelles is specialized structure and function, and therefore nucleus will then be formed. So then we will get a cell which contain nucleus plus other organelles, specialized organelles. So those kind of cells are called the eukaryotic cell because they got well-developed nucleus. So we can find eukaryotic cells in all the eukaryotes. So eukaryotes are referring to organisms which are made up of eukaryotic cells. So over here, we can see that Protoctista, fungi, plantae, and animalia are the kingdoms, right? The kingdoms which are made up of eukaryotes. So those eukaryotes are made of eukaryotic cells. So here I'm bringing you to see the process of so the process of evolution. So on Earth, organisms start off as unicellular organism, which is very simple prokaryotic cell. And as time passes by, so this arrow stands for time passes by, there is compartmentalization. So specialized organelles are formed inside the cell, especially nucleus. So then eukaryotic cells are formed. So it first started with a unicellular eukaryotic organism like amoeba, paramecium, right? So unicellular. Then after that, the cell starts to group up together to form tissue. Tissues become organ becomes system. So the organism becomes more complex. So there's an increase in complexity. That means increase in specialization of the structure and function inside the cell as well as in the body of organism. Because when the cells start to arrange the tissue organ system is actually in the organism, body of organism. So from unicellular organism, it becomes multicellular organism. Okay, so it's very interesting, huh? Right, so this is a table to compare between prokaryotic and eukaryotic cell. So when you're asked to compare means you must state the similarity plus the differences. So when the question asks you to compare, your answer must contain both similarity and differences. Unless the question asks you to state the differences, and then you have to state only the differences. If compare, if you didn't write similarity, you will not get the full marks, because compare means similarity and differences. Okay, so prokaryotic cell has no nucleus and no membrane-bound organelle. So this is the most basic definition for prokaryotic cell. Prokaryotic cell got no nucleus, no membrane-bound organelles. Eukaryotic cell got nucleus and also membrane-bound organelle. So the membrane can be a single membrane, like secretory vesicles, Golgi vesicles, transport vesicles are all single membrane, or double membrane, lysosome also single. Double membrane like mitochondria, chloroplast, nucleus. Okay, as well as organelles without membrane like ribosomes. Right, ribosomes. Okay, there are no membrane. Okay, now what about the DNA? DNA in prokaryotic cells. So imagine prokaryotic cells are the bacterium. So easier for you to remember, bacterium are prokaryotic cells. So the DNA is a circular DNA and it is naked. Naked means the DNA just appear as DNA. There's no additional thing around the DNA, like it doesn't wear any clothing. So that's why it's called naked. Whereas the DNA in eukaryotic cell is a DNA that wrap around histone protein, means that additional things around the DNA. So it's like it's wearing a clothing. So that's why it's not naked. So DNA plus histone protein will form the chromosome. Okay, sometimes we call them chromatin when it's not coiled up. When it's condensed, it's called chromosome. Okay, so DNA plus histone protein is chromatin, as well as DNA plus histone protein is also the chromosome. So the DNA in bacteria is found free in the cytoplasm. DNA in eukaryotic cell must be found in the nucleus. So ribosome for bacterium is always 70S. Now the S is a capital S. Okay, sometimes you learn it as 20 nanometers. Then in eukaryotic cell is 80S, other than mitochondria and chloroplast, which originate from the bacterium. The rest of the ribosomes in the cell will be 80S ribosome, or the size is 25 nanometers. So cell wall of bacteria is always made of murein and peptidoglycan. They're the same thing. Cell wall of plant cell is made of cellulose or lignin, and fungi is made of chitin. Cell surface membrane of bacterium, remember, cannot carry out endocytosis and exocytosis. Cannot carry out endo and exocytosis. Okay, but there are some location of the cell surface membrane that will fall into the cytoplasm of the cell. So eukaryotic cell membrane can carry out exocytosis and endocytosis. Other than cell surface membrane, it will also contain mitochondrion and chloroplast. Okay, size of prokaryote, please memorize, is 1 to 5 micrometer. This one you must memorize. Very easy to memorize because the size of mitochondrion, which is believed to come from the bacterium, diameter is about 1 micrometer. Okay, and the length is about 1.5 to 10 micrometer. So this one, when you memorize, you know that the size of the bacteria is about 1 to 5 micrometer. Okay, size of an eukaryotic cell is 10 to 100 micrometer. Now, 100 micrometer is actually the size of a human egg cell. Okay, egg cell. So the egg cell is larger, right? The size of a red blood cell you have to memorize. You need to memorize the size of the red blood cell. The size of the red blood cell is 7 micrometer. This one, all of you have to memorize, 7 micrometer. Now see, 7 micrometer is smaller than 10 micrometer because red blood cell has no nucleus. So that's why it's smaller than the usual smaller size cells. Okay, so average cell size is 10 to 100 micrometers for eukaryotic cell, but red blood cell having no nucleus is smaller than 10 micrometer. So it's 7 micrometers. This one you have to memorize. Okay, So what is the organism that's made up of prokaryotic cell? It is the bacterium, right? Organism that's made up of eukaryotic cell, it can be unicellular organism, the prokaryote, and the multicellular organism like fungi, plantae, and animalia. Okay. Okay. So now we're going to look at the structure of a prokaryotic cell, means the structure of a bacterium. Okay. So on the right-hand side are the basic structures of a bacterium. So all bacterium will contain this basic structure. And then on the left side are additional structures that may be present in only some bacteria. Okay. So let's look at the structure. Cell wall is present in all bacterial cells and is made up of peptidoglycan or murin. So they call it cross-link because peptidoglycan could be a polymer, okay, long chain polymer, and they're interconnected. So when you say interconnected, means you may connect them together, right? So this is called cross-link. So you go across between neighboring peptidoglycan polymer and you link them together with certain bonds. So this is called cross-link. Okay, cross-linked peptidoglycan. Hang on. So plasma membrane is like the cell surface membrane, cytoplasm, ribosome. So this ribosome must be a 70S ribosome. Okay, 70S ribosome. So you can add this into your notes. Then the DNA is always circular DNA and is naked. Now, what about additional structure here? We have got flagellum. So some bacteria will have flagellum for locomotion. Locomotion means to move. Okay, loco means location. So to move from one location to the next location. So locomotion. Okay, then capsule. Capsule is usually gel-like substance that's surrounding outside of the bacterial cell wall. So that will provide additional protection to the bacterium. Then we have got infolding of plasma membrane. So you can see this plasma membrane here fold inwards into the cytoplasm. So when it fold inwards, it will carry out specialized function here. Even though bacteria got no membrane-bound organelles inside the cytoplasm, but the cell surface membrane can fold inwards to carry out specialized function. So the membrane here may contain special enzymes for it to carry out specialized function. So for example, to carry out photosynthesis, to carry out nitrogen fixation. So the membrane will contain enzyme for it to carry out these two processes. Then mesosome is also a specialized location where the cell surface membrane also fold into the cytoplasm, and it contains some specific enzyme for it to carry out, for example, to help in the formation of new cell walls. Okay, so mesosome is not in your syllabus, so you don't have to learn what is mesosome and what is it used for. All right, plasmid is a very small circular DNA. Okay, this is an additional DNA on top of the circular DNA where all bacteria will have. So what is the difference between these two DNA? Is circular DNA is the basic DNA of all bacteria. They must have it because it is this circular DNA that will carry the genes that code for this bacteria. So therefore, sometimes in the textbook, you'll find that they label this circular DNA as the chromosome of the bacteria. So the chromosome here doesn't mean DNA plus histone protein. The chromosome in bacteria means this is the main DNA of the bacteria. So the other additional bacteria, which is the other additional DNA inside the cell, we call them the plasmid. So plasmid will be smaller than the circular DNA. And usually plasmid is the one that will be carrying the gene that confers antibiotic resistance for the bacterium. So the antibiotic resistant gene will be carried inside the plasmid. And the plasmid can be transferred from one bacterium to another bacterium, which is totally different species. Okay, so how do they transfer the plasmid from one bacterium to another bacterium? They will use pili. Okay, one pilus, many pili. So the pili is a special structure that's synthesized by the bacteria. All right, it's like a tube for it to transfer the plasmid to a neighboring bacteria. So this process, they call it a conjugation, or they call it sexual reproduction. Okay, actually it's not sexual, it's a conjugation that will happen in the process called sexual reproduction for bacteria. So the bacteria will transfer their plasmid. First, they will replicate the plasmid because they want to retain their own plasmid. So they replicate the plasmid. After that, this plasmid will be passed from this bacterium to another bacterium through the sex pili, right? Through the sex pili by the process called conjugation. So this is how, when a bacterium that's antibiotic resistant, it will cause the rest of the bacteria in your body to become antibiotic resistant, because it will transfer the antibiotic resistant gene to other bacteria through this process. Okay. So now we look at the structure of virus, right? Virus. So virus is considered as a non-living thing. So that means virus is not considered as an organism. It's a non-living thing. Okay. Why is it not an organism? Because it does not fulfill all the characteristic or the features that a living organism has. Okay. So therefore, it's considered as non-living thing. So in the notes I've given to you, there's a section which I show you that all living organisms have got at least 8 properties. So viruses have got a few of the properties missing there. Okay. So it's considered as a non-living thing. Now, virus is a very small infectious particle. So the size of the viruses will be between 20 to 300 nanometers. You need to memorize this size, 20 to 300 nanometers. So it's a very small infectious particle. So when you use the word infectious, so what does it come into your mind when you think of this? Resume recording. So when you see the word infectious, right, it refers to 2 things. Infectious is either you refer to the disease or you refer to the pathogen, the organism that can cause the disease. Okay, pathogen. So when you say infectious, either infectious disease or infectious pathogen. Okay, so the meaning of infectious means it is transmissible. Infectious means transmissible. Transmissible means you can transfer it. We use that the word transmit. You can transmit it from what? From an infectious person to an infected person to an uninfected person. So that's the meaning of infectious. So it must come from a person who's already carrying this pathogen. Then this person come to contact with another person which is not carrying the pathogen. And then the infected person pass this pathogen to an uninfected person. So this is called transmitting it. So whenever you use the word infectious, that means it can be passed from one person to another person. So that's why it can be passed to the whole world. Okay, starting from Wuhan in China, right? After 1 to 2 months, already spread to the whole world. So very fast, right? So this is called infectious disease. So the virus is being passed around to the whole world. Okay. So this is a definition of virus. Okay. It has, so these are definition of virus that I'm showing you here. All this. Virus has no cellular structure. So remember we say that the basic structural and functional unit of organism is a cell. Virus is not a cell. So therefore it's not a living thing. So what do you mean by it's not a cell? It doesn't have the composition of a cell. So you know that the cell is made up of the cell boundary, cell surface membrane, and it contains cytoplasm, and it contains nucleus. This is for eukaryotic cell, okay? May not be contain nucleus. For bacterium, no nucleus, okay? And ribosome. So all cells will contain cell surface membrane, cytoplasm, and ribosome, okay? Not necessarily nucleus, okay? But viruses don't have any of this material. So virus is not a cellular structure. So it's not a cell, so it's non-living thing. However, it can replicate inside living cells. So virus is like a very interesting particle, like dust particle in the air, dust particle in the soil, that when it happened to enter a living cell, it will be able to convert this living cell into a viral production factory. Isn't that interesting? Let's say, for example, you've got a factory in your house, okay? You've got a factory of producing, let's say, bread, for example, okay? So suddenly someone enter your house. So we call this someone virus. And then this virus will take over all the machinery in your house. So instead of producing bread, you will start to produce baby viruses. So it has taken over your house and convert all the electricity in your house, the water in your house, the raw material in your house, all the machine in your house, just to make baby viruses. Isn't that amazing? Okay, so that's what the virus will do. It's not living, but it can still make more new viruses. It cannot make. It will make somebody else make for them. Okay, so it can replicate only inside living cells. So therefore, all viruses are parasitic. Now, parasites are organisms that live, right? Parasites are organisms that live. They either live on your body, okay, or they live inside your body to obtain. Why they want to live? Because they will obtain nutrients from your body. So that's why they live in it. And then because of that, it may actually harm the cells, okay, or harm the body to a certain extent. So that's the definition of parasites, right? Organisms that will live on your body, like some fungus that live on your body, right? Some ticks that live on the body of, let's say, your dog, for example, and mites also, okay? So obtain nutrients from the body and harm the cell or body to a certain extent. So virus is a parasitic because they have to replicate inside the cell. So let us look at the basic structure of a virus. So what will a virus contain in the structure? It's not a cell, but it's got certain structure there. So the very important structure it must contain, of course, is the DNA or RNA, because DNA and RNA are the universal molecules that carry genetic code. So if this virus enters a cell and it wants to make the cell make more new viruses, it must carry the code to inform the cell this is how you're supposed to make the virus. So the codes are all carried in the DNA or RNA. So it's called genetic code. All genetic code of living organism must be carried in the form of DNA or RNA. So virus must have got DNA, RNA. So the viral genome will be in the form of DNA or RNA. Okay, so genome means what? Genome means the complete set of gene or genetic material that's present in a cell or in an organism. So the complete set of gene that is needed to make the whole organism is called the genome. So the genome of the virus is inside the cell, Inside the virus is DNA or RNA. And there'll be a capsid. Now, capsid comes from the Latin word. It stands for little box. So you can see here, this box is hexagonal shape. So it's called a little box that surround the DNA or RNA. Now, this little box here, right, can you see it's made up of a lot of spherical structure that join up together to form the wall of the box. So this little spherical structure here is actually a protein. So this protein is called the capsomere. Now the suffix mere, right? This suffix mere stands for parts. So the parts that make up the capsid, the basic part that make up the capsid is called the capsomere. So the capsomere is the protein that form the basic structure of the capsid. So you join all these capsomere together, you'll form the capsid. Okay, just like you join the tubulin dimer together to form the microtubule, the wall of the tubular shape of the microtubule. Okay, so then you've got envelope, right? Okay, envelope. Now this envelope is the phospholipid. It's made out of phospholipid, right? So this envelope is made out of phospholipid. So this is very similar to our cell surface membrane, but we cannot call this cell surface membrane because virus is not a cell, right? Can I call it a membrane? Yes, I can call it a membrane, but I cannot call it a cell surface membrane. So therefore, I call it an envelope. And this envelope is made of phospholipid, all right? It may be present in some virus. It may not be present in all the virus, okay? Then all viruses have got envelope protein on top of it. So there'll be envelope protein, okay? So let us recap the structure of the virus. Virus consists of DNA or RNA, which carries a full set of genes, the viral genome, and it will be surrounded by a protein coat called capsid. In Latin means little box. So this is a protective coat because you need to protect its genome, right? It's so precious. All the genetic code to make new viruses are there. So the capsid is made out of separate protein molecules called capsomere. An envelope, which is a membrane-like outer layer, which is made of phospholipid, may be present in some viruses, not all viruses, okay? And the surface of the envelope may contain the envelope protein, okay? Virus infects the whole cell and injects its DNA or RNA to the whole cell and take over the protein synthesis machinery to make new viruses particle. So when a virus enters the cell, it must make sure that the viral genome, the DNA or RNA, must be injected into the whole cell so that the whole cell will be able to read the viral genome to make new viruses. So it will hijack the machinery of the cell. Right? The machine of the cell means ribosome is used to make protein. Mitochondria is used to make ATP. Right? So now it hijacks all the machine of the cell only to make virus components. Okay, so to make the capsomere, to join the capsomere together, to make the RNA, and then to make the envelope, to make the envelope protein, to make the new viruses only. Okay, interesting, right? So virus converts the whole cell into a viral production factory. So like this scanning electron micrograph here, you can see a lot of the green color particles are the viruses that are released from this wave-like structure is the cell surface membrane of the whole cell. So the figure is showing you the HIV viruses budding off from the lymphocyte. Because HIV viruses, they attack our lymphocyte and convert the lymphocyte into a viral production factory. So the lymphocyte now no longer go around to kill the viruses, but they make new viruses and eventually the lymphocyte also will die. Okay, so isn't that amazing? Okay. So let's look at the sizes of virus. So virus, you know, the range of the size of virus can be very big, right? So it ranges from 10 to 300 nanometers, isn't it? So over here, so this is the size of the thickness of our hair is 500 nanometers. Can you see that? So the virus is actually smaller than the thickness of our hair, so we cannot see them. So these are the various viruses and the size of the virus. So HIV virus is this one here. Okay. I need to add one more, it's a COVID virus, right? So you know what is the size of COVID virus? It's not here, okay? Now, we're talking about sizes of particles and cells. So you need to roughly know the sizes of at least the 3 different types of cells here. So the first one is sizes of eukaryotic cells is about 10 to 100 micrometers. Please memorize this. Sizes of prokaryotic cells like bacterium is 1 to 5 micrometer. And the sizes of virus can range from 20 to 300 nanometers. So all these 3 measurements you need to memorize. Okay, must memorize. Now, the next thing is this. This diagram here actually shows what are the objects that we can see using our eyes. So if you look at this one here, okay, these are the sizes of the object where our eyes are able to see. Now, can I ask you, I want to ask you, if I give you a ruler here, right, a ruler in cm, what is the smallest size of the object that you can see? What is the size of the smallest object that your eye is still able to see? Using the ruler I'm showing you here, can you tell me what is the size of the smallest object that your eyes can still see? Please type your answer in the chat. Frog embryo. Okay, give me the size. I want the measurement in millimeter or centimeter or whatsoever. How many millimeter? Take your ruler and see now. Okay, you said 1 millimeter, okay? So I want you to, on the paper, draw the length of 1 millimeter. Okay, draw the length 1 meter. So here, if this is my ruler, I will draw the length of 1 millimeter is from here to here, right? So which means to say that this is not the size, the smallest size I can see, because the smallest size that I can see is the smallest dot that I put on the paper and still my eye can see, not the line, it's a dot. So let me make a smallest dot here. You can still see, right? And this dot is smaller than 1 mm, you know. But I can still see, right? And you can still see, right? So what is this size? Type your answer. Cannot be 1 mm, right? Yes, you know. You got the answer. So what is the size, my class? 0.1 mm. So the smallest size that our eyes are able to see is down to 0.1 mm. And 0.1 mm is how many micrometer? 100 micrometer. Okay, you need to be able to convert unit like how I convert now. When I see mm, I want to convert to micrometer straight away. I think, ting, ting. In 3 seconds, I can give the answer. Ting, ting, ting means, ting, ting, ting means 3 seconds. So you need to be able to do this working until like this. Okay. So 100 micrometer is the size of the largest cell. That means to say it's an egg cell. So therefore, when you do an ultrasound scan on the ovary, ultrasound scan on ovary, and there will be mature egg cells, you will be able to see the mature egg cell because it's 100 micrometer. Okay. All right. Of course, ultrasound scan already magnified lah. Okay. Even if you don't magnify, you can see because 100 micrometer. Okay. So anything smaller than this, your eyes cannot see. No. So what happened? If your eyes cannot see, but you still want to see, you have to use microscope to see already lah. Correct or not? So that's why we have to use microscope. So anything that you want to see using microscope, because your eyes cannot see. Why like that one? What happened? Okay. Okay, so this is what I want to show. Anything you want to see that your eye cannot see, you must use the light microscope. So the light microscope allows you to see object of this size. Stretches from here to here, visible using the light microscope. Okay, so you know that light microscope allows you to see any object that's bigger than a certain size. So can you tell me what is the minimum size that you can see using light microscope? Qi Jing Yi. Faster. Minimum size you can see with light microscope.
I have no idea.
We just learned only, right, in previous lesson. Okay, Mahi Jaiswal. Smaller size you can see using light microscope.
Is it 200 nanometers?
Yes, very good. This one, oh, you must memorize. 200 nanometer is the resolution of light microscope, right? So the size of the object you can see using, that you can resolve using light microscope, or you can see using light microscope is 200 nanometers. So anything above 200 nanometer, you can use light microscope to see. Now, electron microscope, what is the minimum size of object that you can see or resolve using electron microscope? Gia?
Yes.
Minimum size you can see with electron microscope?
0.5 nanometer.
Very good. So you all need to be able to give me offhand like this. Okay, 0.5 nanometer. So therefore, any object is smaller than 200 nanometer, you cannot use light microscope to see it. So you have to use electron microscope to see. Okay, so what is the sizes, minimum sizes of things that you can see using electron microscope? You can see to the level of not only virus, because virus is 20 nanometers minimum, right? But we can see down to 0.5 nanometer. So we can even see protein structure, the molecule protein. We can see up to DNA structure. So it's amazing, isn't it? We can see down to molecular level, structure of molecules. Wonderful, right? But for microscope, we can see down to 200 nanometers, size of 200 nanometers. So 200 nanometers is how many micrometer? It's 0.2 micrometer. So down to 0.2 micrometer, 0.2 micrometer. We are still able to see bacteria using light microscope. Okay, because the sizes of bacteria ranges from 1 to 5 micrometer, right? 1 to 5 micrometer is actually bigger than 200 nanometer. How do you know it's bigger? Because this is actually 0.2 micrometer. So therefore, you can still see. You can still see bacteria. In fact, if you take the Vitagen or nowadays what you drink, other than Vitagen, I forgot the other one. Yakult, right? Yakult. And nowadays you can see a lot of, not only Yakult, Farm Fresh also got the yogurt drink. So all of them got bacteria that should be alive. So if you take the yogurt drink, you put a drop on the microscope slide and you look it under the microscope, you can see a lot of bacteria that's highly motile, moving around everywhere, swimming around. Okay, you can see the bacteria because its size is bigger than 200 nanometers. Okay, so other than that, the sizes of things that I want you to memorize, please memorize because it will be very helpful, is the size of red blood cell you must memorize. Red blood cell size is equals to 7 micrometer. Now I type micrometer because I cannot write micro, the symbol micro here, okay? And memorize the size of cell surface membrane. CSM stand for cell surface membrane is 7 nanometers. So please memorize this one. Okay, size of red blood cell 7 micrometer, cell surface membrane 7 nanometer. Then what about 10 micrometer? So size of nucleus is 10 nanometer, which is 10 nanometer, no, 10 micrometer, which is very easy to remember, isn't it? Because if a cell, right, contain the nucleus and the cytoplasm of the cell is so thin that you almost cannot see the cytoplasm, isn't it the size of the cell is about size of the nucleus, which is 10 micrometer? Like this is cell surface membrane, so thin it's almost wrapping the whole nucleus. So we got this type of cell in our body. This is example, lymphocyte. So there's a type of lymphocyte whereby the nucleus is so big and cytoplasm seems to be almost cannot be seen one. There the size is 10 micrometer. Okay, so please memorize all these cells. All these cells. Some of them are not cell. Nucleus, cells. Please memorize all these sizes. Okay, must memorize. Okay, so passive question I will not discuss today. I will discuss it in some other days, can or not.